Welcome to the Molecular Moments Podcast.
In today's episode, we sat down with our guest, Dr. Jennifer Zemo, Director of Bioanalysis at BioAgilytix. Jennifer is an expert in protein chemistry and bioanalytical assays. She has excelled in what I consider one of the hardest jobs in the pharmaceutical industry. But you'll have to listen to find out what that is and why I think it's so challenging. She's a leader, mentor, and a scientist that's highly respected in the field of bioanalysis. I hope you'll enjoy our conversation as much as I did. We are talking science as scientists do. So without further ado, here's another episode of Molecular Moments. Welcome to the podcast, Jen. I'm delighted to have you join me today. Can we start with you? Just give us a few highlights of your career.
Hi, Chad. Sure. Essentially, I went to a small liberal arts school for undergrad. I went to Washington College, majored in chemistry. And then when I was looking for grad schools, I stepped on the Carolina campus and fell in love. So I came to UNC, got my Ph.D. in biological chemistry. And then after that, I did my postdoc at NIEHS, looking at signal transduction pathways. After that, I went to Talakris, which is now Grifols. It's a plasma-derived product company. And finally, I ended up at BioAgilytix, where I've been for almost 8 years now.
Wow, we're lucky to have you at BioAgilytix. So clearly you were driven to science. One of the things I like to point out when I talk to other colleagues from BioAgilytix is when I hear that somebody was a chemist, I feel a bonding moment. And I didn't realize your undergrad was a chemist. So you're a chemist like me, turned large molecule expert. So that's really cool. What drove you to science? Like what got you in that direction of being a scientist?
It's a good question. It doesn't really run in my family. My family, they're math people and teachers and things like that. So I used to want to be a math teacher. And then I took chemistry in high school. And I'm like, you know what? I like this. I'm pretty good at it. I think I'm going to go in this path. So once I got to college, I just liked the material. It came naturally to me. And so I just followed that pathway. But, my parents wanted me to be a doctor and I didn't want that path for myself. So I chose the more, still healthcare centric, but more into the details than the overall big picture.
Yeah. So do you think ending up in the pharmaceutical space, was it an accident of where you got your first job, or was it sort of consciously moving in the direction of healthcare and healthcare research?
I think it was mostly consciously moving in that direction. So I knew I didn't want to be in academics. There are very few role models, especially for females in academics. So I knew I wanted to get married, have kids. And so I thought industry would be the best place. And I still wanted to stay in an area where I knew I could help people. And so I think that's what drew me to the healthcare space.
Cool. Well, I'm excited to learn more about some of your research. Certainly one area that is still really important in pharmaceutical development is kinases. And you did a lot of research in kinases in your Ph.D. Maybe tell us a little bit about what are kinases and why is that important to research?
Yeah. So, when I was looking at what type of project I wanted to look into, my PI was actually in biophysics. So I had to do a project in that. But I also was really interested in how the cells communicate, how signals are transduced. And with kinases, it phosphorylates things, right? So, and that's what I tell my friends. I studied a protein that phosphorylates things. And just that one change in a molecule can cause such a huge signal cascade. And that's what I wanted to look at, just something that you think is so small and insignificant in the real life. It can make a huge change in the body. So that's why I wanted to look at it. And I continued that from grad school through my postdoc, is looking at those signaling pathways and what they can mean, because there's so much that's still unknown about it.
Yeah, so I think of a lot of the kinases and the kinase inhibitors in oncology treatment. Is the primary therapeutic area with kinases, is it typically around oncology, or does it touch in other therapeutic areas as well?
I think it's mainly oncology, but I think there's lots of different things that maybe we don't even know about yet.
Cool. And so how'd you end up in your postdoc at NIEH?
So I knew I wanted to stay in this area. I looked at various places and I knew that NIEHS had a lot of core labs, so you could learn almost any technique that you wanted to. So I went there and I learned flow cytometry, gene expression, confocal microscopy, lots of different techniques that I hadn't been exposed to before. But I knew I wanted signal transduction, so that's the type of path that I went down, and they had one whole department just on signal transduction there.
Oh, wow. Yeah. So that sounds exciting and a great opportunity to get exposed to so many different technologies. I think it certainly has helped you be the scientist you are today, right? Because you know so much more than just kind of that narrow space. So that's really cool. So you went from your postdoc and then you were, as you mentioned, Talakris Grifols. Can you tell us a little more about that company? And was that the company that Afshin Savavi was at? Because I recall he was a guest previously. So maybe you can talk about your experience working with Afshin at that point.
Absolutely. So he actually left about a year before I got there. He was the head of the team that I joined before. So we do have a connection that way. And some of my coworkers at Talakris worked for Afshin. So I had heard all about his antics and his personality and how exciting he is about everything he does. But, there I did preclinical and clinical assay development. And it was my introduction to human health and regulations. And, before, I don't think I wrote down lot numbers. I just did the assays and moved on, right? But now you have to write down absolutely everything you do. So I learned that there. It was eye-opening, I think, to see all of the tasks that you need to do to get a drug to the market. So, I was involved with various teams and committees looking at all the things that, we had to accomplish to get something on the market. And it was really interesting.
So it's a big jump to go from pharmaceutical company to a CRO, right? And some people might think you're a little bit crazy. What why do that? What attracted you coming out of the pharma to go into a CRO?
So first, my boss at Grifols, at Talakris, she had come from a CRO. So I had heard about the culture and the pace and the stress level at times. But ultimately, when Grifols bought Talakris, I was laid off. 2/3 of my team were laid off there. So obviously, I knew of Afshin. I had friends that worked at BioAgilytix. So I submitted my resume and I met with Afshin, and it was 1 month from the time that I was laid off at Grifols to I started at BioAgilytix, so a very short time frame. And I jumped right in.
Yeah, and what year was that?
2013.
Yeah, 2013. So you've been with BioAgilytix for a good chunk of the really, amazing growth with the company. Tell me a little bit about how that's been, growing with a company and at the pace that BioAgilytix has grown.
It's been insane but fun at the same time. So, when I started, I think there were maybe 45 people, and now there's almost 600 worldwide, I think. We're still hiring and growing, and I think we've had some growing pains along the way, but with that comes a lot of opportunity for growth. So I started in the lab. I was running assays every day, and then my group slowly grew month after month, and now there are 60 people on my team. So I've just been along for the ride, willing to take on more work and more initiatives throughout the company. And it's been fun. I've learned a lot.
Yeah, there's no doubt. We, consider you as a go-to person in the laboratory. Certainly, from the business development side, everybody wants Jen Zemo in their customer call. So, yeah, I mean the leadership and the success that comes out of the projects that run in your group are, is amazing. And a big part of that is customer service, which means so many different things at a CRO. So in my introduction, I said that I think you have the hardest job in the pharmaceutical industry, right? And I'm going to tell you a little bit about why I think that. So, and I'm mostly referring to the BPM side of that, but then as a manager of it, project managers, that is, bioanalytical project managers, as a manager of those bioanalytical project managers, you oversee and help support them and do that job. But the combination of skills that you have to have between the project managing, the people skills to lead, the scientific skills, the leading by influence skills, right? So tell me a little more about that challenge and about what it means to, customer service, what that means to you in this role.
I think customer service is critical for a BPM role. You can teach science, you can teach assays, you can train people to write and, analyze data, but you can't teach a personality. So you really have to have that fit of being able to communicate well, making sure that you're aware of what your clients need and not just what they want. So asking the important questions and delivering what they need. And so I think it's critical to have customer service training. So we do that here at BioAgilytix. And I think I've worked with some clients for almost my whole 8 years here, and you develop a real relationship with them. I'll meet them for lunch when I go to conferences, or, just ask how each other's children are, or pets, if they get a new puppy. And I think that really means a lot to our clients to know that they have someone they can turn to when they need something.
Yeah, a mentor of mine when I was a project manager many years ago, guy Mark Stiles. You may even know him. He's probably audited BioAgilytix at some point, but he was a senior project manager. This is going back almost 20 years, and he said, Chad, the job is to be the fingers of the customer into the lab, right? Or if the customer were to hire somebody and put them into the laboratory, you need to think, like that's what you are, right? So you're not just obviously sort of, I'll say beholden, I don't think that's the best word, but to the internal group, but you also have that customer focus. You need to make them look good, right? And that's, if you can make them look good, probably in the end, everything else comes together. So tell me, what's a day in the life look like pre-COVID and now virtual? I guess you're probably not in the lab more than a couple days a week now. So how has that changed for you?
Right. So I go into the office once or twice a week just to check in on things, see how people are doing, walk around, make sure they know I'm here if they need me. Before I was in the lab every day and people would stop by my office, ask a quick question, or we'd draw something on the whiteboard if they didn't understand, what a sample pretreatment was or why we're getting specific signal in a certain assay format. And now we have to do that on Teams, and it's much more difficult. It takes longer. So what could have been a 5 to 10 minute conversation is now something you have to schedule for a half an hour on someone's calendar. So I think that's been very challenging. Our BPMs have worked tirelessly throughout the pandemic to make sure they're still meeting timelines. It's been tough for sure. It's been longer hours than usual, but we're still rocking and rolling and creating solutions for our clients. So it's just very different. And I miss that social atmosphere at work. I miss going around and saying hi and asking how someone's weekend was, because it's hard to schedule 30 minutes to talk about that, right? So I miss that aspect, and I hope we get back to that soon.
Yeah, and there's so many new folks that we brought in. I'm sure your group has grown in the last year, and there's probably people you hardly connected with, right? I mean, it's just a reality, isn't it?
It's true. I set up one-on-one meetings with everyone on my team in the first couple of months of the year just to make sure. I mean, there were some I had never met in person. I had no idea what they looked like. So I'm like, turn your camera on. I need to know what you look, just in case I see you in the hallway. What do you look like? And, it's just not our culture to be that way. So we're a very social, teamwork-oriented culture at BioAgilytix, and we do miss that for sure.
Yeah, certainly I expected when I joined BioAgilytix that I'd be on site, visiting and spending time with everyone and interacting and sitting down. COVID has changed so much. One of the things I have really enjoyed about these podcasts is the different folks that I've gotten a chance to sit down with for half hour, 45 minutes at a time, and really have some of these conversations, just see what's going on, and just kind of have something, some casual discussion. So when you bring in new staff and when you think about how to, develop the new staff and the newest scientists, what kind of advice do you try to give the newer scientists that want to develop in the organization? What's some advice there?
Ask questions. That's a big one. Always ask questions, because none of us know everything. So I encourage people to talk to anyone in the company. They don't have to ask me if I'm up the chain of command, as you will, but go to QA, ask questions there, QC reviewers, study coordinators. It's really about getting your name out there, learning the various areas of the company, and then having a senior scientist train you or show you the ropes around the lab. So we have a buddy program. We're going to implement a mentorship program. And I just think it's critical for people to learn about others and rely on the team, because it really is a team environment, and we all have such diverse backgrounds that if someone has an issue, someone else in the company has seen it before. You just got to find them.
Yeah. So tell me more about the mentorship program. I actually have not heard about this, so I'm interested.
Yeah, so we hired at the end of last year a global director for learning and development, and he was my buddy. So we have a buddy system, right? And so we've met a lot, and he just presented his strategy and it was approved. So we'll be implementing a mentorship program for each level in the company, analysts, scientists, BPMs, not necessarily your manager, but someone else in the company who can help you grow and learn in the different assays that you want to learn or the different customer communication techniques that you'd like to learn, and just kind of go through those steps and make sure you're headed on the right path.
Yeah, you've been involved in a number of different new initiatives like that. Another one that you've been involved in is training. Training has been a big part of what you've, I'll just say, stepped up to do at BioAgilytix. So tell me about your passion for training a little bit. What's the story there?
I think it comes from, me wanting to be a teacher a long time ago. I like that part. I love being able to see on someone's face that they get it. You draw something on the whiteboard and you're like, this is how it happens and this is the theory behind it. And then it just clicks. And that just makes me so proud that, they get it. And now they understand and they can teach it to someone else. So for me, teaching someone else or training them, it makes them more independent. It takes time, but it's time well spent. So we want to make sure people understand what they're doing, why they're doing it, and that they can teach someone else how to do it.
What are you working on learning right now? What's kind of your passion to learn more about?
That's a great question. So right now, I'm setting up a summer internship program. So I'm learning about, what types of projects can we have them do? How do I want to present it? Writing job descriptions. I've never had to do that before. So bringing in different aspects of the company and learning about it. But in general, what I like to learn about, besides coaching and training and things like that, is just the various molecules that our clients come up with and why that's important and what novel idea is going to be next. So I love investing some time in looking at people's websites and figuring out what they're going to do with the next type of molecule.
I don't know when it'll be published, but later today I'm going to talk with a couple of individuals from Chimeron Bio that are looking at a new mRNA delivery or RNA delivery mechanism. So talking about new and cool science, that should be a really great podcast. So you'll have to check that one out as well. So when you are looking at new projects, is there kind of a favorite type of assay or something that you just like, this is what I really know, this is what I'm really good at, and you can just dive into it? Tell me about that. I see you smiling. So what do you think?
Yeah, I love ADA. So, I do a ton of PK assays, but at the end of the day, ADA is my favorite. There are so many different types of sample pretreatment. It's not as structured, so you can really get creative with what you do. There's lots of techniques that we haven't tried or that are novel and that we want to try soon. So, it's just being creative in assay development. And I think for me anyway, an ADA assay is where you can get most creative.
So Jen, you and I had the opportunity to collaborate a little bit, or I kind of brought an idea. Well, you brought an idea and I kind of pushed it, and then you came back with a great idea to do this study on biotin interference. So, which was really cool, really timely. I didn't know much about it myself until you had brought it up. So, can you tell us about why is biotin interference a thing at all, and why is it a thing in bioanalytical assays?
Sure. So I actually saw a presentation at WRAB last year, and it was on how biotin may or may not affect assay results. It's common. You'll see it at your doctor's office for diagnostic type tests. We use that streptavidin biotin combination in a ton of different plate-based assays. And I hadn't seen anyone really do it for an ADA or a PK assay. So when I brought that up to our scientific team, we set up a case study. So we looked at various PK assays, ADA assays, MSD versus GyroLab. We looked at ligand binding NAB assays. And at the end of the day, most of the time, there was no interference. But we did see a false negative result in our ADA assay at the low positive control. So it's very possible, if someone is on a high dose of biotin or on biotin supplements for hair or nails or whatever it might be, there could be some effects in results for a clinical trial. So for an ADA assay, you may get a false negative if they are on high levels of biotin supplements. Or we saw in one of our PK assays where it was pretty sensitive assay, the MRD was only 2. We saw some interference and a lower recovery than what we would expect. So, if we're making decisions based on that, then we could assume that we need to dose at a higher level than we really need to. So, my advice for most people would be, instead of looking at all of the different types of assays we have, perhaps include in the clinical trial that people go off of their biotin supplements if possible, so that we don't have to worry about that kind of interference.
Yeah, and I get the impression that most of those biotin supplements and things are probably more of the vanity products that have the really high levels. Is that right?
Yeah, I think the middle and low levels are usually those just over-the-counter type of biotin supplements for hair and nail growth. But I think there may be a very high level that can be prescribed if needed.
Okay. So I wasn't aware of that. I was thinking it was kind of the other way around. See, that's where you can keep me straight. So what, what's next for Gen Zemo, right? What do you think, what's down the road? What else, what are your dreams, your scientific dreams that you might want to pursue or investigate?
Yeah, I don't know. And it's part of my job to think about this on a regular basis, right? Career development. I like where I am right now. I enjoy the science I do. I'm pushing myself more to do more training, more coaching. I want to learn about coaching more people, establishing a summer internship program to help train younger scientists. But in general, I think I'd just like to be overall in bioanalysis. I love assay development. That is my passion. I really enjoy the challenge, especially in a CRO world where everything has to be done yesterday. Developing tough assays is hard, but I've never been one to step away from a challenge. So that's what I enjoy, continuing to learn about that and growing in my career as far as maybe branching out to cell-based assays. So I'm a plate-based person, but I always like to learn, so I'm open for almost anything.
So you're sitting in a room with the management team for operations, and somebody says, we got a super tough assay. Jen, are you saying, bring it on, we'll do it in our group? Or are you saying, we're going to let that go to somebody else? Which one
I say bring it on.
Awesome.
Yeah, actually, a couple of years ago, maybe three years ago or so, we had that happen. So we got a rescue project from a Pharmaceutical Company. They had it at 2 other CROs. They couldn't get it to work. There was a ton of target interference. I was given 30 days to make it work. So I'm like, you know what? Let's do it. Let's get it done. And on top of our other normal workload. So we were doubling up on assays every day, running it. Well, we got it to work. We got it validated, ran a ton of samples, and it's a repeat client. We still have several projects from them. So it was a great opportunity. We had a good time, and I actually got it approved for my team and I to go for a spa day. So we had a great time doing that.
Oh, nice. Yeah.
Yeah.
Work out some of that stress from figuring out the tough
Exactly.
assay.
It was a tough 30 days,
so
but we did it.
So Jen, one of the topics that I'm really fascinated about, and it seems to come up in every one of these discussions I have, is mentoring. Mentors, that we have and the mentoring role we play. And sure enough, it came up in our conversation once again. And I wanted to dive a little bit deeper into that on a more personal level for you. Maybe you want to mention a few mentors that have had an influence on you, or how those mentors have changed and directed your career.
Sure. So in life, I think in general, my mentors are my parents. They always told me I could do whatever I wanted to do. So I think naively, as a female coming into science, I had no idea that wasn't completely normal. And once I got to grad school and realized, there's only 4 female professors in the chemistry department, I thought that was kind of odd. And I just, my parents have always been those who pushed me to do whatever I wanted to, no matter what. And I've just taken that with me, and I push other people to do the same. Don't think about the details or, any of the politics or the drama around it. Do what you want to do and make it your own. As far as, a mentor in science, I think my first one was Dr. Frank Cregan. He was my mentor in college. So, I went to a small liberal arts school. We learned a lot about writing and reading. But on top of that, he really pushed me to figure out what I wanted. And, he was our organic chemistry professor. He taught very differently than most. So everything was group work. And whatever we learned in class, we would then do in the lab. And so you really had to figure out why something was happening, or, what does it mean when you have a nucleophilic aromatic substitution reaction? What color crystals are you going to get in the lab? And so that was really interesting to me to see teaching done in a very hands-on way. And then, when I was applying to grad school, he really pushed me to go for the best. So I applied several places. I had a lot of choices to make when I decided on UNC. And I was thinking about taking the easy way out and going closer to home. That's just the introvert that I am, I think. But, he really pushed me to go visit the different places that I was accepted. And it was narrowed down between Penn State and UNC. And as soon as I walked on the UNC campus, I'm like, I know this is where I'm supposed to be. And when I told him that, he's like, yeah, I told you so. So he's always been one that I've gone back to for advice. And we still keep in touch. When I'm at home, we try and meet up once in a while. I worked for his wife when I was in college in the chemistry department. So it was really a family atmosphere, and I enjoyed that. And then I think, as someone in industry, my supervisor at Grifols, Christina Cruz, she worked at a CRO before. She gave me advice on what to do. She took the time to really teach me about the assays we were running and why they're important and what the data would be used for. And, she was just very interested not only in what I could do at work, but what my family life was like. And, when I told her that I was pregnant with my daughter, she was so excited for me. So it's someone that really, I think, coaches you not only in your career, but also, taking an interest in what you do outside of work and asking you about that, pushing you to go for the stars if you want to. Those are the mentors that I think about when people ask me.
That's super cool. I love that story. And I love the connection that you're keeping with your professor from, some 15 years ago or whatever that was. And because that means a lot to me as well. So occasionally I have folks that used to work for me in previous jobs or things calling me up, asking for career advice. And it feels good because that is certainly, one of the things I want to do is to kind of leave that impact. And I'm thinking, as you're talking, I was thinking, wow, I bet Jen has a bunch of young analysts in her group that are like, would answer the question, say, boy, Jen Zemo would be my mentor. And she's done so much for me. And I think that's something really that you should be really proud of, because I know, like I said, I
hope so, for sure.
Yeah. You're a go-to within the company. And I think, I know a lot of people look up to you, whether they're in the executive team or in the sales team or in your own team, for what a great job you do. So also another thing, we're going to be able to travel again here this year sometime, I think. So what's on your travel bucket list and why?
Oh, I want to go to Italy. I've wanted to go for years. I want to see the culture, the food, obviously, but so much history there. And my husband was a history major. He's really interested in that kind of stuff. And I don't know, it's something that you look back on and say, this was here hundreds of years ago. So yeah, I would like to go to Italy just to have wine with every meal. Yeah. Pasta, all of that kind of stuff.
I'm with you on that. In fact, we've been talking about a trip to Italy for a while. So that's one that's definitely on my bucket list as well. One more for you. What motivates you to work so hard every day? Because I know that you do.
I think number one, because I enjoy what I do. I think if I didn't enjoy it so much, I wouldn't work so hard. But secondly, I'm the breadwinner in my family. So my husband's a stay-at-home dad. He takes care of the kids and they're remote learning. And so I can focus on my career and doing my job. So I think it's a combination of those two things.
Oh, that's awesome. Jen, thank you so much for being on the podcast. Loved hearing about your commitment to training, mentoring, leadership, customer service, all these great things, the fantastic science you're doing. It's cool to see how much the science is still exciting you and carrying on that legacy. This is exactly the kind of conversations we love to have on Molecular Moments. Just want to ask if you would want to throw out there, or say hi to mom and dad or anything while you're on the podcast. Here's your moment.
Thanks. I think my mom is one of the biggest fans on Facebook of BioAgilytix. So hi, Mom.
Awesome. So thanks again, Jen, for being on the podcast. That is all for this episode of Molecular Moments. If you enjoyed today's episode, be sure to subscribe on Apple Podcasts, Spotify, or your favorite podcast app so you never miss a conversation. If you'd like to hang out with us outside of the podcast, we have many webinars and other presentations available for your enjoyment and education. Enjoy them as Jen Zemo's mom does by visiting bioagilytix.com to see what's coming up and how you can stay in touch. And don't forget to keep an eye out for more episodes coming soon. We're looking forward to some great guests. We have world-renowned experts talking about rare diseases, diversity in the pharmaceutical industry, new and exciting technologies, and a conversation with a patient who's benefited from the recent tremendous developments in our industry. Molecular Moments would not be possible without the support of our sponsor, BioAgilytix Labs. BioAgilytix is a global contract research organization specializing in large molecule bioanalysis. Based in Durham, North Carolina, with labs in Hamburg, Germany, and Boston, Massachusetts, BioAgilytix provides high-quality bioanalytical services to leading pharma and biotech companies around the world. They offer assay development, validation, and sample analysis under non-GLP, GLP, and GCP, as well as GMP quality control testing. If you're looking to work with a team of highly experienced scientific and QA professionals through all phases of clinical development, look no further than BioAgilytix. For more information or to speak with their scientists today, visit their website at www.bioagilytix.com.
Thanks for listening to the Molecular Moments podcast.